All right, so this one is all about how eukaryotes regulate gene activity because we don't have those simple operons where we just stick on a repressor and turn off all the genes or, whatever. Each one of our genes is regulated independently of each other. And we have a lot more places we can regulate as well. So we can stop it anywhere along the steps of gene expression. So let's review really quick what gene expression is. And so here's just a quick little diagram. And we'll kind of point out where different things happen and where different regulations can happen. So gene expression is simply taking that code that's in DNA and turning it into some kind of product that the cell can use. I put mRNA. Honestly, there's other RNAs, tRNA, rRNA, all those. They just don't become protein, so they would go and do their job. Okay. The process of making DNA into, I actually probably should have put that up there, but anyway, is called transcription. Okay. The process of taking our pre-mRNA into RNA, to mRNA, is called RNA processing. And then taking our mRNA to protein is called translation. Okay, none of that is new to you. Well, the processing is new to you, but transcription and translation should not be new to you. And so what we're going to look at now is where can we stop this process? Let's say that I start the process because I think I need this protein, and then I realize, oh, no, I don't need that protein anymore. Where can I stop this process? Okay, and so the first place I can stop this process is up here at the DNA. Okay, so DNA is either open or it is clumped, like a hairball. Okay, if it's open, RNA polymerase can get in there and do transcription and allow this whole process to happen. If it's clumped together, this is called euchromatin and this is called heterochromatin, just to remind you of those terms. If it is clumped together, it can't get in here. So this is one way we can stop gene expression, is by altering the way that the chromatin is structured. Okay, so we alter chromatin structure. I don't know why this isn't really focusing for you. I'm sorry, it's a little fuzzy. All right, and so we can do that by a couple things. We can add a methyl group. That's going to take it to heterochromatin. It's going to cause it to kind of clump up together, and that's going to decrease transcription. Okay. We could also add an acetyl group or a phosphate group. Those are going to open it up, so they're going to take it more to that euchromatin, and they're going to increase transcription. Okay, so that's one way we can regulate whether or not we decode our DNA, is just simply by the way the DNA is structured. Okay, another way is we can stop or encourage, if we want, transcription initiation. Okay, I don't know if you remember all the steps, there's a lot. These activators bind to this enhancer region that folds over. There's mediator proteins, there's a DNA binding protein, there's transcription factors that have to come in and bind to that TATA box. All of that has to happen before RNA polymerase can bind to that promoter region. So if we don't want transcription to happen, we don't make the transcription factors, or we don't make the activators, or we don't let the DNA bending protein bind to the DNA and bend it, or we don't let those mediator proteins be there, or we don't, there's multiple ways we can stop transcription. And so if we stop transcription, we never get our RNA, we never get our protein. Okay, the next thing we can do is we can control RNA processing, and the main way we do that is we can use alternative RNA splicing. And if you'll remember, alternative RNA splicing is where I had, let's see. Okay, so let's say that's my gene. From that, or my RNA, from that I could make protein ABC, I could make protein ABD, BCD, I could make protein AB, or protein AC, whoops, AD, BC, BD, CD. I mean, there are all different kinds of proteins I can make depending on what becomes introns and what's exons. In this one, D is an intron, so it gets cut out and thrown away. In this one, C is the intron. So let's say I start this process because I need protein ABD, okay? But when I get to right here, I realize, oh, I don't actually need ABD anymore. Something changed, and now I need ABC. Well, what I can do is instead of cutting out C and making ABD, I can instead cut out D and make ABC. So I can switch which protein I'm getting by changing the way that I splice it. And so that's a way we can use alternative RNA splicing to change the protein. Another thing we can do is we can either stop or cause mRNA degradation. We can degrade it. Because what happens with all of these, with our RNA and our protein, when we're done with it, we break it back down into the monomers and recycle them and reuse them. And so when we're done with an RNA, it's going to be degraded anyway. So if we realize at this point, oh, I don't need that protein anymore, I have enough, or the job is done, or whatever, I can stop making it, we'll just cause this mRNA to be chopped up and recycled because we don't need it anymore. And so that's going to affect the lifespan of mRNA, okay? And I'm just looking through our I can statements. The first is, all of them say, I can describe the way in which eukaryotes regulate gene expression by altering chromatin structure, controlling transcription initiation. And I didn't write all those things, but those were, activators, transcription factors, those kinds of things. RNA processing by controlling mRNA degradation. The next one is translation initiation. So if you'll remember, for translation to begin, let me finish writing that before I talk. Your mRNA comes out, from your nucleus. The small subunit of the ribosome binds, and then the large binds. Well, if you can stop that RNA and that small subunit from binding together, you can stop translation. And so what can happen a lot of times is something will bind to the mRNA. And sometimes that's another RNA. There's those microRNAs and the small interfering RNAs. They can bind to that mRNA, so it can't bind to the ribosome. And so that effectively stops translation because you can't bind to the ribosome, you can't do translation. And then the next one is, let me turn the page, there's so many I can statements, it's on 2 pages. I can regulate gene expression through protein processing. When this protein comes off of the ribosome at the end of translation, it is often just an amino acid chain. Sometimes you'll start having some secondary. If you'll remember, secondary was your helix or those beta pleated sheets. So sometimes you'll have some of that starting, but really they need to be folded completely, and chaperonins do that. You might remember that's a word from last week. But chaperonins can do that, and that's called protein processing. And what it does is it gets that protein in that final structure so that it can function. Because remember, if the structure's not right, the function's not right. So if it's like this, it's not functioning because it's not the right structure. So these chaperonins are going to help process or fold that protein to get it in the right structure so it can function. So we can control that. We can prevent that folding from happening. Also, insulin, if I'm remembering correctly, looks like this, something like that. And you actually have to cut this connection right here. I still don't know why it's not focusing, but anyway. You have to cut this to make insulin active because it changes the structure, so that turns it from inactive to active. And so you can have a bunch of insulin floating around like this, and until you cut it or cleave it right here, it's inactive. And so that's another thing. We can just let it stay inactive until we need it, and then we can go in. And so that's the way we can regulate gene expression. Even though we've done all the steps, our protein's not active, and so gene expression has not been completed until the protein's doing its job. And then the last thing is protein degradation. So the same thing as I mentioned with mRNA degradation. The longer the protein is alive, the longer it can do its job. And so when we no longer need it, we just degrade it. We chop it up into the amino acids and we recycle them. And so we can control the lifespan of the protein that way. Because again, gene expression, we always talk about gene expression as DNA to protein, but actually gene expression is when that gene is fully expressed, which means that protein is acting. It is doing its job. So it's not just making the protein, it's getting the protein to a point that it can do its job or stopping it from doing its job because it's done. We don't need it anymore. And so those are all the different ways. Here's kind of a flow chart, and then all of the locations along that flow chart that eukaryotes can regulate gene expression. Because again, you don't want to make proteins if you don't need them. That's wasteful, and wasteful things tend to die. And you also don't want proteins doing their job if they aren't needed, because that can create problems. So I hope that helps. If you've got questions, let me know.